thank you everybody for being here today. So my name is Flaminia Zane and I'm a Scientific Training Officer at EMBL-EBI here in the UK. So if you've followed some of our webinars before, you may notice that today we have opted for a slightly different format. So what's going to happen is that I will start with a general introduction about what we do at EMBL-EBI and what's a data resource, so that you have some background about the general topic of today. After that, we will move to a series of flash talk. Let's say we'll be joined by some representative of data resources here at EMBL-EBI. They're already in the call, but I will introduce them later on when we will start that part of the webinar, but you can already see their name in the slides here. After that, we will have a panel discussion. So thanks to everybody who already submitted their question before, but we will have time also for taking question live. So as Ajay mentioned, please, if you have any question, just type it in the Q&A box. So let's start with what's EMBL-EBI. So we are part of a wider organization, which is the European Molecular Biology Laboratory. It's an intergovernmental European organization, and its mission is to promote, support, and perform and coordinate research in molecular biology and more in general in life science in Europe. But we also aim to have a worldwide impact. So there are 6 sites at EMBL across different countries in Europe, and each site is devoted, let's say, to a different branch of research in life science. Our site, it's EMBL-EBI, so it's EMBL and European Bioinformatics Institute. So as you may know already, we are specialized in bioinformatics, and we are based here in Inkstone, which is a very tiny village of around 300 inhabitants. So maybe you never heard of it, but we are very close to Cambridge in the UK. I've added a link at the bottom of this slide because a question that often comes up during webinars is, are there any opportunities for internship at EMBL? Or, how can I see if, there are some job openings, etc. So if you click on this link, and Ajay will also share this slide in the chat during the talk, so you can already start to visit some of the link if you like, you will be redirected to a page where there will be opportunities listed across all EMBL sites. So you may find this useful. What are our mission at EMBL-EBI? Basically, they are EMBL mission, but applied to bioinformatics. So one of our main mission is deliver data resources. So that's connected to what we're going to talk about today. And I will come back to the data resources in a moment. We also perform excellent research in life science, of course, with a strong bioinformatics core. We train, as part of our mission, the next generation of scientists. That's why we are here today. We also engage with industry, so we work very close with academic partners, but also with industry. And a very famous example of a collaboration that was born between EMBL-EBI and industry is the AlphaFold database, we were also going to talk about today. And finally, our, let's say, broader mission is to coordinate bioinformatics across Europe. So with this, hopefully you have a overview and introduction of what's EMBL-EBI and what we do at EMBL-EBI. And let's move to the second part of our title, which is data resources. So I just introduced this slide to put everybody on the same page. So what's a data resource? We can define a data resource as a structured and accessible collection of data. So normal data resources include tools, infrastructure, and documentation that enables users to store data and retrieve data. This normally is common to all data resources. And then we can have, in some cases, additional tool to support the interpretation of the data or to support further analysis of the data. So it's, what's commonly called also a database sometimes. At EMBL-EBI, we host and curated 45 data resources, and they span a little bit across all the domain of life science. If you have already been in contact with data resources at EBI, you may already be familiar with this image. So here you can see a little bit the extent of knowledge, let's say, in life science that we cover through our data resources. So we go from the chemical scale, for instance, bioactive molecules or MetaboLights, to nucleic acid scale, so genes, genome-wide data, and RNA. to protein level, cellular level, and organism level. So, for instance, databases that store data related to diseases in human. And then we have a higher level of literature and knowledge management, where all this knowledge is somehow interconnected and put together. What's common across all these data resources is that data are freely and openly available. So you can just go to the website of the resource and start to explore, start browsing, retrieve the data, download them, further analyze them, etc. There is only one exception, which is the European Genome Phenome Archive. So in this resource data are not openly available because there are clinical data stored there. So for privacy issue, data available only upon request. But that's the only exception, at least amongst the one that we have here at EMBL-EBI. So as you can see, there is a lot of knowledge stored here. So there are a lot of opportunities, but there could also be some challenges. If you are new, maybe to research, you never used one, you never used our website, you never been in contact with EMBL-EBI, you can feel a little bit lost. So if you're completely new, let's say to our data resources, and you like to start exploring, maybe you want to look for data, or you want to submit data, or you're looking for training to learn how to use a specific resource, I'd say that our website, the EMBL-EBI website, it's a good place to start. So you can see at the bottom of the slide, I also added the link. So if you click on it, you will redirect it exactly to the same web page that I have screenshot here, that is our home page. And let's say you want to look for some data. So for instance, you're interested in some protein structure data, but you don't know which resource stores this data. So you can click here on the top on services or on find data resources. Both these buttons will redirect you exactly to the same page, which is this one. It's our service page. And here you will have a first hyperlink, which is explore all our data resources and tool. If you click here, you will be redirected to a page that will just list all the data resources available. And I think most importantly, especially if you're new, it will give you option to filter. So for the example I said before, maybe you're interested in some structural data, then you may want to click here on protein, and you will only have to scroll through the subset of resources at EMBL-EBI that are related to those kind of data. So maybe easier to find what you're looking for. You may have, let's say, the opposite problem. So maybe you need to submit some data because you're writing down your research. You need to submit it to a journal, and you need an identifier. You need to store your data. You need an identifier to link this to your publication. And you don't know what's the right resource to submit your data. So a good place to start is this button here on our webpage, Submit Data, because this will bring you to a submission wizard. So it's an interactive page where we have not only the list of the data repositories at EMBL-EBI, but you have here a survey. And the first question that this support tool will ask you is, what type of data do you have? This is all interactive, so you can click on the option that relates to your data, and then you will have further questions that will kind of narrow down the resources that may be good for you, and finally output normally only one resource. And you may start to look into it and see if it's actually the right place for your data. Each resource has also some contact points, so you can also explore that, and we'll go through it later in the webinar. Finally, there may be the case where you already know the resource that you are interested in, but you never used it, so you don't know actually how to do it. So we do also provide training about resources at EMBL-EBI. That's exactly what we're doing today. But if you're starting from our website and you want to find which kind of content we have, scroll through our catalog, then the easiest would be to click here on training on the top, or on train with us. Again, this will redirect you exactly to the same page, which is our training website. This is our homepage. We always have some feature courses on the homepage. Normally it's one webinar. So in this case was the webinar that we are currently doing. And we also have some courses that are featured, normally the ones that are open for application. But that's not the only training we have. And the easiest way to start going through our catalog is to use our query bar here on top. So let's say, for instance, you are interested in learning more about Ensembl. So you can query Ensembl, and you will have result page where everything that has Ensembl in the title or in the overview of the training material will be outputted. And once again, what's important is that you have filtering option because we provide training across a wide range of formats. So we have webinars like the one we'll do today, so live training, but this webinar will also be recorded, so then will be available on demand. We have online tutorials and collection that are always available on demand on our website. So if you're looking more for some written content that you can go through at your own pace, maybe you want to filter for online tutorial and look for this specific kind of content. Or maybe you're interested for longer or larger, let's say, format kind of training, so our live courses, and then you may want to filter on the live training side. But that's, let's say, where to start and look for different kind of training related to resource. So you just, the same way you Google, you query through the website. Okay, so with this, hopefully you have a general introduction of what we do at EMBL-EBI, the kind of data resources that we have, and how to use a little bit our website. So now I will move on to introduce our speaker for today. So the image that I showed you before, as you can see, is quite big. We have a lot of data resources, and we're not going to go through all of them today in details. We have decided to focus on the left part of this page. So to talk today more about resources that store chemicals type of data, nucleic acid, and protein. For this, we are joined by 6 representatives of 7 resources. And we will get started by following exactly the same order. So for the chemicals branch of life science, we are joined today by Jane Blackshaw with scientific data engineering at ChEMBL, and Noemi Tejera Hernandez with scientific database curator at MetaboLights. Each of them will briefly introduce the resource, and then we will move on to the nucleic acid side our data resources. So we will have Luis Mirabueno, who is Outreach Officer at Ensembl, and Sandy Rogers, who is Web Platform Project Leader at MGnify. And finally, we'll move to the protein side. So we have Aurelian Luciani, who is Project Leader for Web Development at UniProt, and Paulina Magagna, who is Bioinformatician at the Protein Data Bank in Europe and AlphaFold database. So she will talk about both the resources. After their flash talks, so after their brief presentation, we'll move on to the panel discussion. And we'll get started now with ChEMBL. So, James, the screen is yours.
Okay, thank you. So ChEMBL is a way to explore compound-target interactions across datasets. And so what we do is we accept experimental data from people with chemical assay datasets to share. We also regularly curate the literature and include information from a selection of core journals. So those papers automatically go in. So for compounding ChEMBL, we link to the bioassays performed on it, the activities found within those bioassays between the compound and the target, and it includes diverse assay setups and readouts. We've got everything from whole animal studies to PROTAC studies to multiplex data sets where there are multiple experiments being run in the same well. So the benefit of ChEMBL is that all of the compound structures are normalized to a canonical ChEMBL one. So when you are searching for data about a compound, it doesn't matter how they may have chosen to represent that compound, provided it's accurate. When we receive it, it gets normalized to that. So everything can go through, this is the compound you're looking at, this is everything that relates to it, here's the activities, or this is the target, here's the compounds or the activities or the studies related to that target. You can use the web interface or you can use the API to get this information, and we regularly run training workshops on how to use either of those.
Thank you, James. So the next one is MetaboLights.
Hello, everybody. I'm Noemi. I'm a scientific database curator in MetaboLights. which is a public repository for metabolomic experiments and derived information. And when I say metabolomics, sorry, I include also lipidomics and exposomic studies. So since metabolites represent the downstream biochemical end products, when we compare with other disciplines like proteomics or genomics, the analysis of the metabolome of an organism is going to be the closest, most likely the closest association to the phenotype. Our resource serves as a study repository, so enables the metabolomics community to share findings, the data, the protocols, etc. And a primary requirement for submission is to provide raw data, an open source version of the file if it's available, also information regarding the annotated or identified metabolites, and very important, rich supporting metadata that describe the study, the samples, the assays, and the metabolites, of course. So our resource use the ISA tab, delimited data model, aligns to the FAIR principles, is open source, open access, all the information can be downloaded and used. It is cross-species, cross-technique, captures all metabolomics data across the entire workflow. Where possible, the metadata is captured using control vocabularies and ontologies. So at the moment, we have more than 2,000 public studies in metabolites that represent more than 300 terabytes of data. And there is a significant proportion of human data, but there is other species that are represented. The reuse from metabolized data takes several forms, and it's important to highlight the importance, the value of public and fair data. We collaborate with other resources, with academic institutions and different consortia. And I think it's important to highlight that we are one of the partners of the Metabolomics Hub, which is an initiative that has been created to standardize open data practices between repositories. And of course, we are also involved with different outreach and training activities.
Thank you, Noemi.
Thank you.
So we'll now go to Ensembl. Yeah, Luis, you can tell me when you're ready and I will play the video.
Thank you. Yeah, you can start the video. So hi,
Thank
everyone. My name is Luis, and I'm here to talk a little bit about Ensembl. For those who are unfamiliar, Ensembl is a genome browser that lets you visualize genes, regulatory elements, and DNA variants. And you can also use it to search databases of genetic information to link genomic features to biological function. Ensembl has been around for over 20 years, so it started off with gene annotation from the human genome. But today you can explore annotation for over, I think, 35,000 different genome assemblies. So for those who are familiar with Ensembl, you might notice that the video playing on the screen there is very different to the Ensembl that you are used to. And this is because this year we are transitioning to a new Ensembl website. And this new website is designed to be more efficient with dealing with the growing number of genomic data that is being produced every single day. Thank you.
Thank you, Luis. I'll play the video up to the end. Okay, so we can move on to MGnify with Sandy Rogers.
Thanks, Flaminia. I'm afraid I don't have quite such a multimedia experience, but this is MGnify, and we are EBI's metagenome data resource. So you can perhaps work out from the name that the MG in MGnify both stands for metagenomics, but also hints at magnification, because we are the resource for all kinds of microbial life. So MGnify starts with publicly available samples from a wide range of biomes. So I guess uniquely for these resources, we're built on top of another resource. So MGnify inherits data from ENA, the European Nucleotide Archive. And we take genomic sequencing data from samples that have been submitted there and then analyze them. So we take the raw reads and then we either analyze those directly or we assemble them into assembled metagenomes. And from those, we run a series of pipelines to look at things like taxonomic profiling of the microbes in them, as well as functional profiling. So right now, our database consists of well over 5,000 public studies, so well over 500,000 individual samples. As well as the taxonomic profiling that we do and make available, we assemble some of these where we can, and then we analyze those assemblies. That really helps with getting functional information about the species in there. But we also take these assemblies and predict protein sequences from them. So one of our other data products is a very large database of predicted protein sequences. So about 2.5 billion unique protein sequences in there. So this kind of differs from other protein resources in that these are just predictions from metagenomes, from wild environments. But this kind of greater scale that you get from this can be useful for things like training protein structure prediction models. As well as that, we bin up some of these assembled metagenomes into metagenome assembled genomes, so individual species like draft genomes. And this really helps look at kind of microbial dark matter, so the kind of microbes that are very difficult to culture. So we have a sub-resource called magnified genomes, which includes well over 500,000 annotated genomes, which you can find on our resource. And like many of the other resources, all of this information is freely available on our website or programmatically via our API, or you can download files from our FTP server.
Thank you. We can move on then to the protein side, starting with UniProt with Aureliano Luciani.
Yes. Moving now to the protein section of that talk. So I'm Aureliano Luciani. I'm a project lead in the UniProt team at the EBI. So UniProt is short for the Universal Protein Resource. We are a comprehensive, high quality and publicly accessible resource of protein sequence and functional information. So we're gathering all the known protein knowledge in the world and gathering all of that into the UniProt databases. We actually have multiple databases. The most important of them is the UniProt knowledge base, which most people know, but we also have databases of proteome, clusters of proteins, and also archives of proteins. So in the UniProt knowledge base, we have about 200 million entries, but we also have more than 1 billion unique sequences in the archive. So what we have are primary sequences, including sequence of isoforms, physiological protein function, including subcellular location, pathways, reaction, interactions, and development indices, sequence annotation of domains, PTMs, mutations, disease variants, and much more, as well as structural information, including topology and access to 3D structures from PDBe or AlphaFold DB, as well as other known or predicted structures. We also provide data analysis tools such as BLAST and multiple sequence alignment. And an important thing that we have is also cross-references to more than 180 other resources. So that would be other resources at DBI, but also outside of DBI. So that makes UniProt a good hub in order to explore, from the point of view of protein, different resources. So go back to the genome part of it, go to the interaction section. We are a good hub for that with those links to nearly 200 resources. And UniProt is a consortium. Part of it is at EBI, so this is where I work, but we also have colleagues at ChEMBL in Switzerland and at PIR in the US. And I'll hand it over to Paulina for structures.
Thank you. So we move on to PDBe and AlphaFold with Paulina Magagna.
Thanks. Yes, I'm actually going to talk about 4 different but united resources. So these 4 resources that I have here form a connected ecosystem that we have for protein structure data. So first of all is the protein data bank in Europe. So this is the European founding member of the Worldwide Protein Data Bank, or the WWPDB. So we deal with all the data that has been submitted in Europe, and is essentially a repository for all the actual data that was used to build these protein models. And it contains experimental determined 3D structures for proteins, DNA, RNA, and their complexes. And on the other hand, we also have knowledge base, so PDBe KB. So this is more of like aggregated data that is coming from the different PDB entries that we have, and we organize them by proteins, and it provides you functional and biophysical annotations. But instead of looking at like one experiment, you see a like UniProt centric view where you can see all of the PDB entries that have been determined, obviously, from UniProt. And it, again, aggregates the data from all these different experimental data on binding sites, phosphorylation site, effects of mutation, and also energetic frustration. So it's more of like similar to UniProt, but more for protein structures. And then we also have AlphaFold database. So this is a collaboration with Google DeepMind and EMBL-EBI, and it contains the predictions of protein structures using AlphaFold. And we covered nearly every known protein in the UniProt database. And as part of the AlphaFold database, we also have the confidence scores. So these are the PLDT and the PAE for all of the entries, and they're very, very useful to evaluate the predictions. And lastly, so we have 3D Beacons. So 3D Beacons is not a database, it's a network. So it acts as a single point of entry to find protein structure and data that's coming from experiments or it has been predicted. So it links to data from different providers like AlphaFold database, PDBe, Swiss Model, and Model Archive, and other repositories and annotations. So the key detail here for 3D Beacons is it's gonna scan all of the members of the databases that we have, and it's gonna tell you how many experimental structures are there and how many predicted structures you can also find in just one place.
Thank you, Paulina, and thank you to all the representatives of the resources that have just provided the introduction to their resource. So I hope that with this you had, like a general idea and of what each resource, at least the ones that we have today here, contain in their database. And we will now move on to the panel discussion. But just before that, I'd like to say that apart from this slide, the speaker also put together some extra documentation. So there are some useful links about the specific resources, contact point, etc. We're not going to go through these slides during the webinar because it's mostly just link kind of documentation, but we have put them together and they will be shared with you, and they're part of the presentation that you will receive. So for the panel discussion, I will now stop sharing my screen so you can see all of us better. And I can see we had a few questions already in the chat, but I would maybe just get us started with one question that was pre-submitted by one of you that is now attending the webinar. And it's about how we can somehow modify submission. So if we have submitted to one of the data resource, and so our data are there, or maybe the data that are submitted by someone else and that are already stored there. So someone has asked if can external system programmatically push derived annotation, prediction, or validation feedback back into EBI resources in real time, ideally through a defined API or submission protocol? Or is any update mediated by a curator or a human, let's say, working at the data resource? So for this question, maybe James from ChEMBL can start.
Sorry, audio is a bit rough. That was one about manual curation, yes?
Yes, exactly.
Yeah. So it kind of depends on your resource, because I know that PDBe have got a sort of semi-automated deposition utility, but for ChEMBL, the manual curation is a key part of the quality of the data. So we've got a data structure that is in some ways very defined in that you need to have an STF chemical structure, you need to have unique identifiers in your data set, but in some ways is very flexible in how you can choose to structure it. So although we do not test the experimental validity and quality of the data itself, we do check the structure, and we do check that and things like identifiers that you've used for external resources are correct. So in that case, any sort of automation there, we're going to be having as the first step in before you send it to us, here is a way to validate your data and go and correct your mistakes, rather than we're going to automatically and kind of unthinkingly ingest the data, sorry, which you could do if you have a very, very clear definition of what each data point must be. But if you're allowing any user flexibility, it's not really possible.
Thank you, James. Unless any other of the speakers would like to add something on this question. If so, just let me know. Otherwise, I saw that some of you booked, let's say, some of the questions that were asked. So I will now take one of the questions that was asked just now, that is regarding metabolomics, so for MetaboLights. So one of the attendees is saying, I'm focusing on untargeted metabolomics. How can I get the EMBL chemical library?
Yeah, I will say that there is no such as like a complete EMBL chemical kind of a database for metabolomic studies, like a annotation file that contains all the chemicals that are present in our resource. But what you can do is like for each study, the experiments that have been performed are present in the meta files that are called assays. And each assay is linked to the MetaboLights annotation files, the map files, as we call them, where you find the list of compounds that have been annotated, or the features, because in some cases the compounds are not completely identified, especially in untargeted metabolomics. But yeah, you can like download all this information. If you you can do search for studies, sorry, and I will recommend you can download these metadata files, the raw files, everything from each of the studies. But if you're planning to download the whole database, I will recommend for you to contact us. So contact the help desk. It's easy to find our email and MetaboLights website, and contact us, and one of our software engineers will help you to have all this information. It's possible, but it will be for each of the studies that have been submitted to the resource.
Thank you, Noemi. I'll take now another of the live question that Aurelian would like to answer. So one of the participants is asking, how does one access one specific protein and interact with it? I'm kind of new to this field and would like just to know more about it.
So it depends a little bit on what information you have as an input, in the sense of if you have a name of that protein, you might be able to even Google it, and multiple resources might come up, UniProt being one of them, but maybe you have related information in PDBe or Ensembl and other resources. So if you go into the UniProt website, so uniprot.org, and search, there's also a search bar there. You can also search for that gene name, protein name, or some specific identifier. Or especially if you come with from the results of some analysis before, and you have a specific ID that might be from another resource. I don't know, let's say you have a RefSeq ID and you want to get the information that we have in UniProt for that. We also have a ID mapping service. So you have your specific ID, and so you remember those 180 plus resources, cross-references that we have. Basically, we have all of them connected. So if you come with ID identifiers from the resources, we will be able to link that to the knowledge that we have in UniProt. And once you have found that protein, then we'll get all the information. You can also look for that protein based on the sequence. We'll be using BLAST or peptide search for this, but you can also find that based on the sequence. And then interacting with it, I'm not sure in what sense you mean interacting, but we do have also interaction information in there. I don't think you meant it like this, but we have information about intact or complex portals, or link even. You can have that information in PDBe also. I know they have complexes there. And so there's a lot of knowledge. So I would encourage you to just browse the page from the top to the bottom. It's categorized by function, domains, interactions, and all of that. And so once you have found the information, sorry, that protein of interest, you will be able to explore the information we have, and also see where in that proteome, in that protein, specific information might be related to, if it's at the beginning, at the end, and all of that. So yes, I think if if you want to follow up on that question with a more precise question, I can also answer a bit more precisely, but that was quite generic.
Thank you. So I saw there was another question that was asked kind of 2 times by the same person, and both Sandy and Aurelian would like to take it. That is related to MCP server to query from Claude to our data resources. So in general, and then a specific question for UniProt.
I could continue. I'll also lend that to Sandy because you wanted to say something about that. And I know there are some different initiatives around MCPs within the EBI. So at the moment, we don't have an official MCP server for that. We're working into doing that. But it's true that this is a new way to access our data. We're looking into implementing that in a way that would be most useful for users. But I want to remind everyone that basically all of the data is not only available through our website, but also through programmatic APIs. We have specific training for that. And I would encourage you, if you are able to, if you have that a bit of knowledge of development or even just a bit of scripting, to explore our APIs. That works, I guess, for all the resources at DBI. The best way to access our data at the moment would be through the API. That's the most reliable way. It will be also the nicest way. We don't want you to go through all of the pages and scrape our data. This is not really nice for our servers. We have servers dedicated to that. We have APIs dedicated to that. So I would recommend you to have a look at the programmatic access offerings that we have, and then watch this space for official MCP announcement, especially for UniProt. I know the teams have a bit more to say about that. I don't know, Sandy, if you want to add up to this.
I think you said it pretty much perfectly, Aurelian. That I think most teams, or many teams I know, are looking at this and prototyping it. There are, let's say, unique constraints in operating services like this at an institute like EBI with the amount of data that is available, especially with how quickly these tools may need to scrape across a large amount of data. So it's not as simple as you might think to just put these things into production. So they will be coming, I'm pretty sure. But there's a whole load of interesting constraints which we could go into, but it's probably not really the place.
Thank you. Yeah, there is a connected question that, yeah, Aurelian, I think already kind of answered it, but I think there will be an agreed answer, let's say, across all the different data resources, because someone is asking, is there an AI search engine at the moment that can scrub the database, I think, and generate predicted data based on a query that is linked to EMBL? So that's a little bit, I think, convoluted in the way it's asked, but I guess the main point is, if I'm using, let's say, an AI, can I go and scrape your databases and take this data? Would this be an approach that is, let's say, feasible, or there is something about it? Do you have a take on it? I think Aurelian already said something, but I see this coming often, so I thought of taking this question in case someone would like to add something on it, or experiences that maybe you had. Sandy?
I can add one point that I think is related. That so a lot of these resources, we haven't kind of gone onto the right-hand side of the set of resources that you saw at the start. But on that right-hand side, there's a bunch of kind of knowledge bases and literature services amongst EBI's resources. And there is interesting work being done over in those services towards sort of building knowledge bases that are queryable in, I guess, new ways across data from all of the different resources. So like trying to connect graph networks that encompass knowledge from all of the different resources. So I think that might be part of what is being asked there, is can you connect data between these resources? And that is a kind of, basically there's LLMs behind that to try and join up these data resources into knowledge bases. So yes, but not quite out there yet. So watch this space, I would say. Follow the news section on the EBI website.
Thank you. I think James wanted to add something in this.
Yeah, so this is our semi-regular word of warning for scraping ChEMBL for anything. We guarantee as much as possible that the data that is going in matches our definitions for what should be in a ChEMBL entry. We do not guarantee the experimental validity of that data or the quality of that. So if you pull studies, they may have chosen to annotate or describe things in different ways. They may have different levels of error in their results, and they may not be interoperable. There's a known thing for anybody who's downloading lots of stuff that's related to similar targets, but whereas you can assume that the links that we've got in ChEMBL are going to be accurate, the activities against the compounds, that is going to be no more curated than if you'd pulled all those off PubMed. So when you are looking at a data set to train any sort of model on, I advise looking very carefully at what that resource promises with regards to where it's curating, where it's accurate, and where those boundaries are. Because, for example, anything we pulled in from external link outs is only as good as that particular external resource data.
Thank you, James. Anybody else from speaker that would like to add something on this question, or we'll move on to the next one?
Just going to add a bit of a word of warning, because we already started receiving some user requests about people trying to access our data through LLMs or things like that. So don't blindly trust what the LLM outputs. It will, if you ask it, give me the sequence for that, it will absolutely give you something. Just check where it's coming from. It might just have invented it just because, well, that's it. It will tell you yes and gives you something. So just ensure that it will either pull that data from a web page or from an API, and that will give you a bit more certainty of that data. But absolutely double check the information that the LLM gives you, because it will give you information and it might hallucinate.
Thank you. I think Luis.
Yes, I just want to add to that as well, that a lot of the resources work as versions. So make sure that you check which version this resource is, sorry, the answer is referring to, because some resources get updated every 2 months, every month, every day. So make sure you know the version.
Thank you. Okay, so I see there are many questions coming in, so sorry if I slow a moment down, it's just to go through all the different ones. I see there is one related to training. So someone is asking if we provide financial aid for bioinformatics trainings at EBI. So I can reply to this for the training team and the courses that we organize together also with the resources. I know some resources organize some courses independently, so then if you want to add anything, please feel free to raise your hand and add to what I'm going to say. So as for the training that we deliver at EBI, we do have two different kinds of trainings. So we have the on-demand training that it's available on our catalog, where I showed before. This training is free for everybody. You don't even need to have an account on our website to read through our courses and watch our recorded webinars. We do have also, the webinars, the live webinars are free to attend for everybody, just exactly as this webinar that you're doing today. All our webinars have exactly the same registration process and maybe different formats, but host on Zoom and free to attend. As for the live courses, that's a little bit different, as those normally are open only to a restricted amount of people, and there is an application process and a review process from a scientific committee. And for those, there is a registration fee for successful applicants, and we do provide financial aids. We cannot provide it for all the attendees of our courses, but if you are interested in applying for one of our live courses, you can find those. I can link it after in the chat, the ones that are currently open. And during the application, you can state and you can give reason behind why you're asking for financial financial aid, and this will be reviewed by a committee and possibly supported if your application is successful. Okay, Luis, would you like to add something?
Yes, so we at Ensembl, we also provide training directly with institutes and universities. We can do these both in person and virtually, and there is some help, especially for countries from low and middle income regions. If you are interested, you can email us directly. I've put I think it was an email address in the slides. You should see them once they're shared with you. And we can also customize the workshop according to your interests. So if you have students that are more interested in the clinical side of genomics, we can focus on human. Or if you are, a plant scientist, we can focus on plants.
Thank you. I've just sent a link of the courses, the one that I had mentioned before. And if nobody has anything to add on this, I'd like to take a question that I think is related to what Luis just said. And because you mentioned plant scientists, and one of the submitted questions was, how are the data resources at EBI applied to my field as a student in plant breeding genetics and genomics?
Yes, so this is a really good question, but it's also quite a broad question. So there are, it depends on what you want to study. So in Ensembl, we have a lot of crop species for which you can retrieve the gene annotation and, variants from. One thing you could do is to, for example, compare related genes across species. So you can transfer findings from model plants to crop plants where a bit less information is available. In the breeding context, you can start from a quantitative trait, loci, or GWAS region for traits like yield or stress tolerance, and then check which genes or variants fall inside these regions and focus on a smaller set of promising genes that might be interesting to test experimentally. You can look at SNPs or other variants near those genes and download surrounding sequences, design genotyping assays. You can use the variant effect predictor to find out what variants are most likely to alter the gene function. So it really depends on what your research focus is. But there are a lot of different applications for Ensembl. If there is something in particular that you are interested in, I just want to highlight that our help desk is really, really useful. So if you have a specific question or a specific experimental design that you're working on, we can help you directly with that as well on the help desk. Again, the email should be on the slides, which you will receive later.
Thank you. I saw there was a question that was asked now and was also voted by other two participants, and it's related to metabolomic studies. But it's my feeling that this is more a research question, because the person is asking if we need to produce triplicates of samples for metabolomic studies. So I work with Drosophila. This is my field. Should I use biological replicates in triplicates? So I don't know if Noemi, you want to add something about this that could be related to the database as well, or if it's
I can get started, and
okay.
then yes. So from the metabolomics point of view, I understand. Some samples are unique. So we have some studies where there is one sample is not the regular. And in fact, in those cases, the submitter usually has to contact us to be sure that there is no mistake. Because it could be like, for example, a natural product extract, very precious standard, any sample that for whatever reason there is only one, injection, one biological sample. But from the point of view of analysis, even if it's the case, I will also always go at least triplicates in the number of not biological, but in the technical replicates. So I will definitely, if possible, if the volume allows it, to analyze the sample 3 times or more, if possible. But yeah, I understand that some samples, for the reason, for even because the organism, the type of organism that you're working with, you only can have one. You always can like look for other ways to improve the amount of data that you have. So maybe anybody else want to like say something else about it?
I think we can move to the next question. We have a little bit less than 10 minutes left. So what maybe, I see Sandy would like to take one of the questions. So we read this one first. So I want to run an exploratory data project with my coding group. Which permissions do we need to get?
Yeah, I can take that. So across almost all of the EBI resources, data are freely available, open access for any purpose. In the data licensing information you can find on the EBI website, you'll find there's a commitment to moving towards a CC0 data license as much as possible, which means that's like the most open these things can be. Not all resources and all data types have or can be moved to that specifically, but in general, almost everything is open access and freely available for any purpose. So you don't need permission to access and use the data from almost all of the resources.
Thank you. There is then one question that is specific for Aurelian. So this is part of the question for Aurelian. I started to work on protein data recently and I'm still new. I've seen that UniProt offers access to different databases. UniProt KB seems to be the most complete, but in my case, I found things on UniPark. Could you go quickly through the characteristic of the other database availables, like UniPark, for example?
Just to keep it short, because for the interest of time, if you want to follow up on that, there's also some help pages about those on the website, and you can also contact us and we'll be happy to develop a bit more. But basically, within UniProt, you have UniProt KB, but you also have UniPark. So UniPark is UniProt archive. In there, we store all the known sequences in a unique way. So any sequence that appeared in a publication somewhere, at least once will be stored in UniPark. And then depending on if it's been seen in human or in chimpanzee, for example, it will all be under the same entry in UniPark. So this is the entry archive. It's basically our entry point for sequences within UniProt. And then for some of them, they will be, let's say, promoted, or at least they will enter into UniProt KB. UniProt KB will have sequences, but separated per organism. And in there, we will add annotations, either created by curators, so that would be added manually, or through automatic processes. So in there, you might have more complete information, especially functional information or cross-references. But we also have just recently started to add some automatic annotation also in UniPark. So basically, if you found your sequence in UniPark, you can get a bit of information there, but if you find similar protein in UniProt KB, that might be helpful for you, because that might be the same just with a small mutation, and maybe that's enough for you to do your study. But yeah, they're kind of related. One flows from the other, but if you want to get more information, yeah, please get in touch with the contact form, and we'll be happy to help, especially in your specific case. If you have a specific question on an organism or a specific sequence, we'll be happy to help you.
Thank you. I saw there was one question that was quite voted for ChEMBL, but James is already writing the answer, so I'll let you finish typing. And I'll take the next one, which is, I'm currently working on in silico aptamer designing. Is there any tool or database to identify which protein residues complements with nucleotides? If any of you know if in any of your resources this tool is available. Okay, no problem. So maybe for this question, we can look if in other resources maybe that are not represented today, and we could come back to this later or tomorrow in the email that we will send to everyone. Let me have a look. Okay, so we have 3 minutes left. Oh, James, I see.
Yeah, I can quickly just cover that question verbally as well. Again, as I've probably said a few times, consider the scope of a resource you're looking at. For example, if you look at UniProt, you can see a quality rating for a structure based on if it was manually curated, etc. And at ChEMBL, we don't do that for the data themselves. So we are a data set that confirms these data existed, were published somewhere or given a DOI or submitted to us, and were valid to go into ChEMBL and did not have any glaring errors in the described biology. We are not a data quality reviewer. Similarly for UniChem, the other resource that I manage and am involved with, it is a directory that links identifiers between databases. Nothing in UniChem has any opinion on the quality of the link outs there. So we aim to be as broad as possible. We do not aim to set a particular purpose for the data or a subjective opinion on quality, because different users will have different requirements. So yeah, you have to choose your own filtering. You have to use your own judgment for what is within scope for your research.
Thank you. So we have 1 minute left, and before we conclude the webinar, I like to give some space to all of you. I mean, if you have something to highlight, maybe any update that is coming in 2026 in your resource, or one, I don't know, special advice that you like to give to users of your resource. So if you have anything of this kind that you'd like to highlight as we conclude the webinar. Sandy, we can start.
I'll pick one thing. Lots of exciting things coming. I'm going to pick one, which is for our amplicon analyses of metagenomes. We are about to release support for amplicon sequence variants as part of our analysis outputs. This is something that we've heard lots from the community will be of interest, and that is just coming online at the moment.
Thank you. Luis, I think was the second to raise your hand.
Yeah, I touched a little bit on this during the introduction talk, but Ensembl is releasing a new website this year. So we're going to slowly transition from the current website that you're familiar with to a completely new website. It'll be much faster, especially for those who are in the US. You might have noticed some connection issues lately. This is because all of our power is going into the new website. So please keep in touch on our social media, on our blogs. We update you there whenever there's new data coming up.
Thank you. So Aurelian, I think, was the next to raise his hand.
Just so quickly, if you browse our website, you might have seen the announcements in messages across the website, but we're doing some changes into the selection of proteins we put into UniProt KB. As I said before, all of them are always in UniPark, so they will not disappear out of nowhere. But by March or April of this year, we'll be removing some entries. Basically, we've reorganized a bit the knowledge space that we have in UniProt KB, so that we have a better representation of the tree of life. So some of species were underrepresented and some were overrepresented, especially when they are really well studied. Like E. coli, you might have thousands of entries for the technically same protein, but just in different strains. So we try to reduce that while augmenting the coverage of some of the less studied entries, which means that it might change how you access some of your data if you're already accessing that. So I encourage you to read the blog post that is linked in there just to see how it might affect you. And contact us so that we can help you access your data still.
Thank you, Aurelian. I think Paulina wanted to add something.
Thanks. Just quickly, for the protein data bank in Europe, we have new pages now. So we have more of like a tab view that you can get different data in different tabs. So it's like easier to navigate. And from the AlphaFold database, we actually made an update last year, and we updated to UniProt, but that was last year. So it might be a little bit out of date now. But we are doing more updates for AlphaFold database. So just stay tuned to the social media.
Thanks, Paulina. Noemi?
Very, very quick.
Yeah.
So promise. But yeah, basically last year we have many updates in MetaboLights, and I just want to mention that this year there will be more, so keep your eye peeled. And something related submissions, something that I think an advice I will say if somebody's like thinking about submitting to our resource in particular because of the amount of metadata that we collect, please allow enough time. So do things in advance. Don't try to rush submissions the last minute just before you need to submit your paper. So that's my advice. Thank you.
Yeah, that's a good point that I think can apply to all data resources in general. For personal experience, when I was working in research, I applied a little bit last minute and wasn't the best idea. So I learned from my mistakes. So unless anyone would like to add something before we conclude, there is one last thing I'd like to highlight, and is that we work very closely in a training team at EBI with all the data resources, and we support them to do outreach and training. So often when a data resource launches a new update, a new version, there is a new feature, we run a webinar together, or there could be updates on the online tutorial and quick tours that they have on our catalog. So often a good way to also get updates on what's happening in the space of the resources and what's new is going through new webinars that we have, and in general our training catalog. And we have a few webinars that will be coming soon from UniProt. So I will be providing link in the email that you will receive tomorrow to everything that is already open to registration. But watch the space also our training website for everything that that's coming up in the next year. So with this, I'd like to thank all the speakers that have joined us today for also staying a little bit past time and preparing a presentation to show to you today. And also like to thank everybody who has joined. We had quite a full room and plenty of questions. So I hope this was useful and you have now kind of a mental map on what we do a little bit on EBI in the space of the data resources. And you know where to go if you're looking for something or if you are looking for help. So thank you again. With this, I'll conclude the webinar and see you in next webinars this year.
Thank
Bye,
you.
everyone.
Thank you. Bye.